Yeah, so my name is Maria or Masha. I go by Masha. This research was in the context of cancer, but I believe it's a great modeling strategy to use also within the context of neuroscience. So using predictive preclinical modeling, basically using machine learning to improve personalized medicine predictions. So just a little bit about cancer. Cancer is an accumulation of mutations that happen to accumulate, and then this exponentiates into cancer proliferation. So it's not just one mutation, it can be very many different mutations in very many different cell types, so they're highly heterogeneous. And what we were trying to do is use data from cell line cultures. So these are in vitro models where different drugs were screened onto these tumor samples. And there are several databases that have this drug sensitivity data. And like, say for one has 60 cell lines and around 50,000 compounds, whereas another one has 1,000 cell lines and around 260 compounds. So heterogeneous data, also coming from different databases. So we were trying to use this large-scale data to develop the drug sensitivity prediction algorithms. So basically this middle picture kind of conveys what we're trying to do. There's a pre-existing tool called GDSC tool, and what it takes is an input of a tumor genetic profile and then the drug screening data that I mentioned previously, and uses that data to predict the best pharmacological treatment for the given tumor genetic profile. So the features that are in this genetic profile are expert selected. They're SNPs, so single nucleotide polymorphisms, just a single mutation in a gene, copy number variants, and methylation. And this is just an example of how the tool would take in that data. Genomic features is a binary matrix, and the drug response is continuous values. And this is an example. Well, let me first just say that this tool takes different types of analysis, ANOVA, so that would be pairwise comparisons between a genomic feature and a drug response. And it also uses regression models like lasso, elastic net regression, and ridge regression. And it uses k-fold cross-validation to determine the best parameters for the models. And so this is one example of the type of output that came out of this tool. So this is looking at one drug 1047, and along the x-axis are different mutations, which are features. And so let's say on the very right, there's the TP53 mutation, and the red indicates drug resistance. So here you can see that if a tumor has a TP53 mutation, this is an indicator of much higher resistance to that drug. Whereas on the very left, the CTNNB1 mutation confers a little bit more sensitivity to that drug. So using something like this, but on a much larger scale for all of the drugs, we can give the tool an individual patient's tumor and kind of figure out, okay, let's eliminate these drugs. They probably won't work for you, but some may work better for you. So personalized medicine. And so there were multiple end goals for this research. This was conducted in a government lab, so we were working with a pharmaceutical company to possibly determine which drugs are more important to study, and personalized medicine, and we want more data for better predictions. Yeah, so that's the gist.